Several years ago, the LACEWING trial was designed to evaluate the value of the addition of a FLT3 inhibitor to azacitidine versus azacitidine alone in patients with FLT3 mutant disease newly diagnosed who were considered unfit for intensive chemotherapy. Now, the standard at that time was azacitidine alone. The study originally included a third arm of Gilteritinib alone, but over the course of accrual, Gilteritinib, as we know, was approved for the treatment of relapse and refractory disease. So the study design was somewhat compromised. The third arm with Gilteritinib was removed, and the results of the two remaining arms, Gilt-Aza versus Aza alone, were recently published in Blood earlier this year. Overall, the main endpoint of the study was overall survival, and there was no statistical difference between patients receiving Gilt-Aza versus azacitidine alone. However, we did see markedly improved overall response rate of about 58% in the patients getting Gilt-Aza versus 20-something% in those receiving Aza alone. And there was a suggestion in post-hoc subgroup analysis of particular benefit in patients with FLT3 ITD and those with high ITD allelic ratios with the receipt of the Gilteritinib plus azacitidine versus Aza alone. Now, that study was clearly flawed by not only the changes in the study design, but by the approval of Gilteritinib, with many patients in the azacitidine arm preferentially getting subsequent FLT3 inhibitor therapy. What it did show us, though, is that Gilt-Aza is a great backbone on which to potentially build triplet studies. Recently, we've had a publication of a Phase Ib study looking at the feasibility of Gilt and venetoclax with overall high response rates in patients with prior FLT3 inhibitor therapy. And again, these studies have served as the foundation for triplet studies that are currently ongoing.